Thanks.
Okay, thank you so much. We are looking forward to your talk, Sofia. The stage is yours.
Okay, thank you. Um, so as you probably know, I'll be talking about epibiotic fauna, animals that we can find, or cetaceans and sea turtles, and how to use them as indicators of their host. I have been already introduced, but I'm Sofia. I do my PhD at the Marine Zoology Unit in the University of Valencia in Spain. And yeah, let's get to the talk. Sorry. Okay, so just to have a heads up of what epibiotic fauna is, I'm referring to any animal species that it lives on whales, dolphins, sea turtles. So they have this intimate, sometimes more oftentimes long, like lifelong interaction. And these animals can be associated to these hosts in different ways. They can be parasites, they can be commensals, neutralists, but I like to represent the interactions like this because it's not always really clear what the nature of the interaction is. So some species that have been classified as parasites are not always parasites, or it's not really clear to define. In the case of cetaceans, referring to whales, dolphins, porpoises, um, epibiotic diversity is a bit lower, but it's over like 50 species, so that's quite impressive. For sea turtles, it's about 400 species, animal species that we can find on them. So they are basically floating or swimming ecosystem. Starting with cetacean epifauna, we can see like it's quite diverse, but we can see that there's one group that is particularly abundant on sea turtles or species rich. And these are cyanids. This is family of amphipods. We have like about 28 species, and they're quite host specific, meaning that each species would usually be found on a single or maybe two cetacean species. And what is really interesting about them is that they are transmitted by direct, so physical contact between the hosts. So that makes them really interesting as a tool to study host interaction. And I have a nice example of that. First, we find them here. So that's a striped dolphin. We usually find them on the blowhole. And on dolphins, their abundance is like their intensity of infection is like very, very low. But then we find cases like this. For instance, this stranded whale. You know that humpback whales are like black in color, so dark gray or black in color. But we can see that this one is like brown. And what happens here is that it had a lot of cyanids. And what happened to this whale is like 3 days before stranding, it was found entangled in a fishing net. Uh, some divers rescued the whale so it could swim again, but fortunately, 3 days later, it died because of the starvation, because of being in the net for that long. We could collect a lot of cyanids of the species, the Amos boobies, and it was really interesting to see that there were thousands of them and they were still alive. So it was quite impressive to see them crawling and moving around. Yeah, quite impressive. And as I say, they are really host specific. So we usually find or basically find this species, the Amos boobies, on humpback whales. And as we can see in the map, the blue points are records of this cyanid species of humpback whales, and we find them like all over the world because humpback whales migrate so much. But we can see an interesting thing here. So we see 2 points that are not blue, they're red and yellow, indicated these are two other cetacean species, a right whale and bottlenose dolphin. So that raises a question of what is happening here. So there's this interesting note about this. And we are super lucky because we have the author in the online audience. Um, so this paper found that Siamese puppies was also found on southern right whale. So that was indirectly indicating that these two species of whale have physical interactions in this area in Brazil. And just this morning, Tammy, the author, just forwarded me this image of evidence, direct evidence of these interspecific interactions between a humpback whale and a right whale in another part of the world, in Australia. So this is an interesting example of how parasites, ectoparasites, can be used as an indicator of host interaction. I'm going to focus more on barnacles, not only because I love them, but they're also really interesting tools for knowing more about the host. We find about 10 species of cetaceans, and as I was saying, they can use as indicators of many different things. But I'm going to give you two examples. Just another heads up: in the case of sea turtles, as I was saying, epibiotic species are much more abundant. We have many, but as you can see, barnacles are also a big component of their epibiotic communities. As we can see here in yellow color, these are all barnacles. We see that in all sea turtle species, we can find barnacles, and they compose a big part of the abundance of epibionts we can find on them. Just some brief introduction about barnacles in general. They can be found in almost anything in the sea. So you have probably seen them already, like attached to ship vessels, any kind of debris that has been like washed up ashore, intertidal rocks, mussels, crabs. So they basically attach to anything they can to feed from the water currents that are generated. And there's an important thing that we need to know about them in order to use them as indicators, and that is that they reproduce by releasing larvae to the water, to the ocean, and they release huge amounts of thousands of larvae. And these larvae have about 10 days, 1 week to find another host to attach on. And this is how they are transmitted. So it's quite a miracle, actually, that they exist, because if we imagine a microscopic larva in the middle of the ocean trying to find a host without really swimming structures or anything, it's quite impressive that it eventually happens. So we can use this piece of information for knowing that where barnacle larvae are released is where they are going to colonize other hosts. So by knowing where this reproduction is happening, we can know where cetaceans or sea turtles are going to recruit different barnacle species. And we're gonna see like an example of this. We studied this on Antarctic minke whales. In Iceland, you have common minke whales, slightly like the smaller version of these. So we have this congeneric species in the southern hemisphere, and we got access to a lot of pictures of the epibiotic fauna of Antarctic minke whales that were collected in Antarctica during the last whaling season. And this was the first time that the epibiotic community was described as a species, because before, minke whales were considered a single species, but now we know that there are at least two species, northern and southern hemisphere one. So we first described the epibiotic community, but we found something a bit strange with this barnacle. This barnacle is called Xenobalanus globicipedis and is usually as beautiful as on the left picture. But what we saw in our sample is that they looked a little bit more like this. So they were degraded. So for sure, they were all dead before collection. And we were wondering why, because this barnacle is found in any cetacean species worldwide. And we were wondering whether it could be the effect of polar temperatures, so really cold temperatures in Antarctica, which were kind of affecting this barnacle. So what we did is, using a data set we had already published, we plotted georeference points of records of these barnacles, records of surveys that were done on cetaceans, where they reported either the presence or the absence of this barnacle, and we plotted this, and we modeled this relationship. So we modeled the likelihood of occurrence of this barnacle with minimum sea surface temperature for all these worldwide records. And we found a really interesting pattern, which we found that the likelihood of finding Xenobalanus was way higher in this range of temperatures, which correspond to this latitude, so tropical and temperate latitudes. So this, among with other tests, allowed us to say that we can use the presence of Xenobalanus in whales that are found like polar grounds to indicate that these whales have migrated from the equator. And this is quite interesting because whale migration was thought to be a quite strict pattern, but now it's been seen more and more that it depends on the species, it depends on the subpopulation, it depends on individuals as well. So it's quite interesting to kind of get to know more about their migration patterns and even distinguish between migrating and not migrating stocks. And I have another example of barnacles being used as indicators in the case of sea turtles. This study was done using samples from the Western Mediterranean, where our university is located. And we first uh, described infection parameters of different barnacles. We found 13 barnacle species, but that was more or less already known. We found this information by using samples from turtles that had stranded in our coast. So we had no information of why they're stranded or where they were coming from. And we also gathered samples from sea turtles that were caught accidentally by fisheries. So these were a really interesting source of data because depending on the fishery that was responsible for the incidental capture, we could know a little bit of where the turtle was located before being captured. So for that, it's important to know that we have like two main groups of fisheries in the Western Mediterranean. So we have neritic fisheries, trammel net and trolling, and we also have now a bit less, but we used to have a lot of pelagic longline. So I'm going to be plotting turtles that were captured in neritic fisheries, so near the coast or closer to the coast in yellow, and those that were captured by pelagic longline, so in the open ocean as blue. What we found is that the epibiotic community of these three species, which are also the most abundant ones, differed. The turtles that were captured by pelagic longline, the oceanic fishery type, had a much greater abundance of these two lepatids, these two species of barnacles, whereas the ones that were captured by neritic fisheries had a much higher abundance of this other barnacle, Chelonibia tessinerea. And we cross-validated this with diet data, so we studied the digestive content of the turtles that died, and we found the same patterns. So these two barnacles, Lepas and Conchoderma, on oceanic turtles, Chelonibia on neritic or coastal turtles. And just by using the composition of these two species, including presence and abundance, we could kind of discriminate with a over 80% success rate whether turtles had been captured by oceanic or neritic fisheries. So barnacles alone were doing quite a good job. What we also did was to measure all the barnacles, so almost 17,000 barnacles from 200 sea turtles. We measured all of them and we used already published growth curves to estimate their age, so basically the time since attachment on the sea turtle. And by that, what we could do for each turtle is to identify like clusters, like age clusters for each barnacle species. So we could kind of infer how many visits, in this case with leopards, to the oceanic um, domain they had been doing in the last month. And just by finding continuous age values, we could also have a rough estimation of the duration of this visit to the oceanic domain. We could also do the thing with Chelonibia and get information about the coastal environment. So for illustrating this, I'm going to give you an example of one turtle in particular. On this turtle, we found really large Chelonibia barnacles, so the ones that are recruited in coastal waters. And we found a really precise gradient of sizes, so ages, on this turtle, decreasing from a size that indicated attachment a year and a half before capture to a size that indicated an age of about 1 month and a half. So basically, we could estimate that this sea turtle had been in neritic habitat, so in a coastal habitat, for about a year. Then the largest barnacle, Lepas barnacle, that indicated oceanic residency, was about the same age as the smallest Kelonibia one. So this kind of indicated this transition from a neritic to an oceanic site. And then we also found a really nice gradient of sizes from larger ones, about a month and a half old, to really small ones. So indicating that they had attached on the turtle just before we had access to this turtle. So by this, we could kind of reconstruct this movement, these ecological patterns of the sea turtle, of course, with some range of error, but in this case, it was a really good estimate because this turtle had a lot of barnacles and the size gradient was really fine. So we inferred this transition from neritic waters about a year before capture, and then to oceanic waters about a month, a bit over a month before capture. And we predicted that the habitat of capture or death of this sea turtle was an oceanic ground. And this was confirmed because we checked our database, and this turtle came from a pelagic loneliner. So this shows that we can reconstruct in a rough way movement between neritic and oceanic domains in sea turtles, which is particularly interesting for the sea turtles that strand in our coast and we have no information of. So just to wrap up, I think the biggest take-home message here is that, and something that would make me really happy is that we kind of switch the algorithm a little bit, that we now know how epibionts and barnacles can inform us about their charismatic host and how useful they can be. So instead of finding this as the first result, like how to get rid of them, remove all the barnacles, we find something like this, like ecological indicators and cool species. So thank you so much for attending, and I'll be happy to take any questions.